please, I am not a plant physiologist, so maybe I'm going to say, I'm sorry. I was saying that I am not a plant physiologist, so maybe what I'm saying is a variety of an insult to you. But, but the plant is fixing nitrogen, no? And then if you leave the roots there, the nitrogen is going to stay there, so you are not losing anything. And you have the plant, the top of the plant, I mean.
No, we were talking with
or you eat the roots. What is it?
Well, the problem is there is now people, I don't know, some people have a lot of time to do crazy calculations, but now they are calculating the amount of nitrogen and carbon that are exported by Brazil to China. Why? Because in grain, I'm talking about grain legumes, which we eat the leaf and we eat the grain. Right? Because in Africa, they love the main salads are the cowpea, black bean, common beans, and they make really nice salads out of that. So what is happening when you transport, I mean, you harvest the grain and export it, you are exporting also all your nitrogen.
Oh, I see.
Because you see, something will remain. Actually, you can plant maize and not add too much nitrogen after a legume. Because, like you say, the root will decompose and the nodules. But the point is that you are exporting. Argentina also is exporting a lot of, not only nitrogen, manganese, magnesium, iron, molybdenum, and so on. So somebody is calculating that it's quite a serious transfer around the world of soil elements. So eventually, the nitrogen fixation will not be enough because you have to add many other things. So maybe become more costly than adding the original NPK. It's a problem.
People is now talking about the root microbiome, and that the root microbiome is required for the health of the plant. So that means that each plant, and actually each genotype of a plant, recruits certain species and certain types of bacteria that help them to withstand not only nutrition from the soil, but to be more resistant to drought and diseases and so on. So people is now trying to figure out which are the really beneficial bacteria and assemble consortia that will not only help the plant to fix nitrogen, but also to be more prepared to defend against diseases. What do you think about that?
Yes. Yes. This could be solutions, and companies in Brazil and in Argentina are already selling this consortia of not only the nitrogen-fixing bacteria, but also the P-solubilizing and all this, and some bacteria that can improve water stress because they make the transfer vessels wider. And the idea is if you add this biofertilizer, 10 to the 9, at least, bacteria per gram of the product, you outcompete. So when the plants germinate and the seed and the roots come out, you will see a lot of the good ones, and they will remove by quantity the bad ones. So this is a good approach. But how many companies? What I'm afraid is I saw in Africa, and I was scared to death, the proliferation of all these small, homemade, university, laboratory producing biofertilizer. And what they do, they take the soil or the nodule, they scratch it, they put it in there. So who knows what that replicate? When I'm talking about Rhizobacter and Fertimax and all these companies, there's an Uruguay, a good one, because they have strict controls. I mean, the government, they have to return the lot, the biofertilizer for checking in very good molecular biology laboratory. So that we can be sure, but how many companies like this one are around the world, especially in Africa or in other places. So that's the proliferation. And if we eat, if they take the seed, I mean, it's a transfer of diseases, which is really worrying me. People have money, and so they can do it. They say, it's a Colombian scientist, say, countries are rich because they make science, good science. They are not making science because they are rich. You see, you understand, because a good knowledge and a good support system are the one. So I will not have any problem with Brazil and Uruguay have company and Argentina, but I really worry about the rest of the world. Okay.
Thank you, Monsignor Sanchez.
Sorry, I want to ask two things that are in relation. The soya, because Argentina is one of the more important exportators of soya. So the soya have this problem also.
Soya is the only grain legume that have been bred for transgenic. You are planting transgenic plant, and some of them are,
This is the second question.
virus resistant to the soybean mosaic virus.
Yeah.
The problem is now they are facing Argentina is 300 virus actually going around the world infecting soybean. So they made it for one virus, which was originally in Argentina the main problem for soybean production. But now they are really scared because many other diseases are coming. And especially coming from people that import. You won't believe it, but people bring, I got a little ball that got a very nice flower, and see, I got it in Singapore and I grow it in the balcony of Buenos Aires. But this one is virus infected, and the insect come, and suddenly you create a pandemic.
Also in the bacteria transgenic.
Yes, yes, they increase the why. I mean, Brazil and Argentina were using the transgenic and close to 6,000 kilos per hectare. The most that you can get in Africa is maybe 1.2. So it's 6 times larger, but they have been breeding for higher nodulation, higher nitrogen fixation, and also some transgenic plants but because you allow, I mean, I don't think they close the, you cannot close, the government cannot close their eyes and say, no transgenic. No, some may be bad, but most of them are good. So,
Thank you.
So they are double transgenic.
Eddie?
About the soybeans, I saw in Uruguay a poster that they were growing rhizobium nodules on soybeans, and they added the bacterium. So I wanted to ask, how do you put in the bacterium? Do you put it on the earth, or what do you do?
There are two technologies, but Argentina use mainly liquid fertilizer, because the good one is the peat-based. But peat is forbidden in most places, except for Canada. Peat is this soil-made fossil soil, so it's not renewable. So they are forbidden in many places to extract. So they switch. So what they do, you have a pot, a plastic pot, huge one. Actually, your machinery in Brazil, what I saw, I would like to drive one of that. So you see the plow. So the machine put the seed and just squeeze a milliliter of the bacteria counting 10 to the 9 cells per ml. You just, that's all. That's all what you need. Yes, because the bacteria start calling, and also they were selected, but this is bacteria that was selected, because like Luis said, you can put the wrong one and the plant will not allow. The plant will not recognize it, or the bacteria will not recognize the root-excreted compound, which are very complicated. It's a dialogue. It has to both parts, it has to be recognized, otherwise they will not be married forever. If they made a mistake, the bacteria, the rhizobia, can become a parasite. And the Vatican's, I'm not going to start more details on that. So, about bad marriage and divorce.
I have a comment or a reflection more than a question. Evolution is supposed to improve properties, to improve adaptation. And you mentioned that plants do not distinguish colonization of rhizobium from pathogenic bacteria. It amazes me that they don't have a specialized mechanism to start a relationship with rhizobium. And also the fact that plants that are symbiotic with rhizobium are more susceptible to viral infections. It should be the opposite. Because they are fixing nitrogen, they should be stronger.
Yes, no, they're not fixing, they're fixing nitrogen. But you see, the people, usually scientists are blaming women. It's a housewife responsible for all the outbreak of virus infections. Because we started to think, we wanted lettuce not too hairy. We wanted, root beer, very nicely round and red. And we want huge cucumbers. So when you modify that, most of the virus-resistant genes were lost during domestications of all these plants. So now the plants used to excrete, you go natural plant, indigenous plant, they don't smell so nice. Sometimes they smell bad because this is insect repellent. And sometimes they are hairy and the leaves are very tiny. They have a lot of things that when you think about a soybean, they doesn't resemble. So most of the resistant genes were lost during domestication throughout the years. So what's happening now, they are putting back these genes, but these genes have a cost on production. It's like, I'm really sorry because I'm talking in the very dangerous, not my comfort zone of genetic, but it's like a box. You can put a certain number of genes. If you took ones, when you put it back, and now you have to change it for another gene, something has to be. I don't think it's space for everything. So usually production, tons per hectare, goes low. Now you have a plant resistant. So it's not so easy.
Rafael and Juan Carlos, and then we close the session.
Mine is very short. But in 2016, there was a very extensive report of the US National Academy of Sciences about genetically engineered crops. And this touches all the different dimensions of the problem. And I will just concentrate in the human health issue, which is what I understand the most. And over 25 years of utilization, there is zero evidence of human health problems. This does not rule out many other types of problems that may bring, but in terms of medical issues, there is, in 25 years, zero evidence of any damage to health. Yeah, yes, just to make
it's stupid, to believe all
and
this
also
nonsense.
a really very good report.
For your presentation, it was very interesting. I'd just like to make a comment first. You mentioned that you have to be careful with the vegetables, and lectins derived from vegetables are known to be absorbed in the stomach, and they kill dopaminergic neurons in C. elegans, and is now believed that this lectin could be the cause of some Parkinson's disease. And so we have to be careful what type of lectins we eat. And the other is more a question. The viral propagation within the plant in general is through plasmodesmata. So do you know if this biota affect the plasmodesmata?
The what?
Plasmodesmata.
Yes, this I understand. But what affect the plasma?
If the biota that's not healthy affect the plasmodesmata.
Well, no, not all the virus. Plasmodesmata are the channel,
Channels.
like tunnels,
Tunnels.
open tunnels between the cell wall, which is the only difference. Most of the virus, they have a gene to open. They even make it larger. But the DNA can freely move also.
Right.
And RNA, virus can be RNA singular double strand, DNA singular double strand. There are even a virus. I was born Gemini, and I love this Gemini because it's two particles like that. And part of the replications, you need to combine these two. And this, the DNA is the only one moving. It's the capside is left behind. So I mean, this protein. So that's what I said. Maybe the plants can be virus-enriched in proteins because they're really leaving behind a lot of this capside.
Okay, Maria Luisa, thanks again.